Welcome, everyone. This is the second annual WashU Bold Pioneer Award ceremony, and really excited this year. We have a fantastic guest speaker. Just a few words about this award. So I think most of us recognize that transformational advances in medicine rarely come with incremental science. Sometimes it requires bold new ideas that challenge prevailing views. And that's what this award is all about. It's about rewarding junior investigators who take that bold step and propose something that is truly outside the box. I'm not going to say too much about this award other than this award was conceived by Dr. Randy Bateman, who of course is a bold pioneer himself. And Randy is the, what are you, Randy? Randy is the Charles F. and Joanne Knight Distinguished Professor of Neurology. And he's director of the Silk Center and principal investigator of Dominantly Inherited Alzheimer's Network and its trials unit. So Randy, I'm going to let you introduce this award, and we'll very excited to hear this talk.
Okay, so my job is to tell you about the award and in the context of what it means and what we're trying to do with the award. So the Bold Pioneer Award, the concept here was to be different than most other awards. Most other awards are given because an individual, a group, or others accomplishes something of merit. And it's the accomplishment that the award is given for. So it's the end result, objective, verifiable, some kind of progress that's been made. The Bold Pioneer Award is actually about the person. It's about the behavior, the actions, the intent of an individual who in this case is in all cases for this kind of award is meant to be a junior investigator who has exhibited some bold, pioneering activity, behavior, action. So we had a conversation last night at dinner about what the definition of boldness meant. And between the 6 people, none of us, a mixture of MDs, MD PhDs, and PhDs, none of us could agree finally if there is a final definition of boldness or pioneering for scientists, because it's really different for each individual. It manifests in different ways. It may come from different sources, from different drives. But there is some commonalities that one can identify about being bold and pioneering. One is that the action and the behavior has the potential to substantially change the way things are done currently. It has to truly be different. In all of our NIH grants, we had to write the word innovative. Your work has to be innovative, and you had to defend why your work was innovative. Scientific progress does not depend solely on innovation. Sometimes being very uninnovative and just making progress counts. Pioneering and bold were put together because it was meant to be pioneering out in front, ahead of others, doing things that others are not doing. Bold was meant to indicate something risky, dangerous, maybe, something that the individual is taking some personal responsibility, some personal risk for, where others might not do it. They might not say that's not likely to succeed. That's not likely, that could go badly. That could end up badly for me or for others, and so I'm not going to take that risk. It's about taking risk. And so each year that we've had this award, we've sent out notices, we've gotten applications coming in, dozens to over 100, and a group of people has to sit down and review and go through these applications. And that committee debates what bold and pioneering means. We have heated debates about, why we're rank ordering the way we're rank ordering, who is deserving of this award. And this year was no different. We got in lots of arguments, and it didn't get physical because we were on Zoom, so it was all okay. But there was consensus that what Claire's work has been and what is being proposed is truly bold and pioneering and has the potential to completely change how things are done in science and medicine. And I think that was one of the bases for her selection. I also want to comment that the intent of the award not to reward success, which most awards do, but to reward those individuals who are taking a risk at perhaps the most delicate stage of their careers when they're getting started. So we strictly limited this not to senior people, not to established people, where maybe it's easier to be bold and to take risk because you're established, you have some backup, but to encourage people new in their careers, people who are getting started, who are trying to establish their programs, to encourage them to be bold and pioneering, where perhaps it may count most. And to entice this, the award gives, I think it's $10,000, personal money, not research money. We all get money for research, and that's a good thing, and we all strive for it every time. But we wanted this, recognizing that this was a junior investigator, we wanted to recognize that individual for taking the risk, and frankly, we don't want them spending it on research unless they really feel compelled to do so. We would rather they go be rewarded personally for their risk and their actions. And the last thing I'll say about the award is it's not about the winner. This award was designed to be about everybody else who might hear the message of the award, who might see that it's valued and recognized and understand that institutions and groups and investigators value those kinds of contributions. And so as much as we recognize and applaud Dr. Clelland and her exceptional, bold pioneering work, we hope that this message gets out and that others hear about it and they're enticed by it and they understand that this is valued. And so for each of you, either listening online or here in person, I hope you go tell everybody you know who could be bold and pioneering at a junior investigator level about this award. I hope you all start gunning to get it next year and the year after. I want you to see a lot of applications in future years and hand this out. But with that, I'll turn it over to Dr. Miller, who will talk about the winner.
Okay, it's my honor and pleasure to introduce Claire. But before I get there, I do want to recognize Brooke Swinney and Tracy Erdman, who did a lot of work to put this together. You can imagine there's a lot of moving parts, and I was following very few of them, so thank you to Brooke and Tracy. Also want to recognize the award committee. Randy mentioned that it is an energetic committee. It's Dave Holtzman, Randy, Risa Sperling, Rudy Tanzi, Rusty Gage. So those are the people that are involved in picking this, and want to recognize them. And me. Yes, I'm also a part of the committee. So now to introduce Claire. So Claire Clelland, MD, PhD, so she's a physician scientist, the John D. French Alzheimer's Foundation Endowed Chair at UCSF in the Department of Neurology. She did philosophy as well as biology at the University of Portland, a PhD at UC San Diego. Also, this was at the Salk Institute and the Cambridge Center for Brain Repair, so she was in the UK for part of this. She had a Marshall Scholarship, a Jack Kent Cook Graduate Fellowship. She went on to medical school at UCLA, residency and fellowship at UCSF. And I've been following Claire's work, which her bold and pioneering work on CRISPR therapies, which will only be a small part, and that's not why she got the award. Thinking about how to use CRISPR therapies for, in particular, C9orf72, bringing energy, creativity, innovation, boldness to that approach. And I was just telling her that I predicted that she'd be in people 5 to 10 years, 2 years ago, that it was a long way off. I think they're closer than that. And a number of groups are following the same steps to think about how we could use CRISPR therapy in the central nervous system, and there's lots of hurdles to that. And maybe some of those hurdles and some of that, well, how do we test new therapies? How do we develop the therapeutics? How do we test them in humans? That led to the really interesting, bold pioneering work that we're going to hear about, thinking about studying therapeutics in people who are brain dead. So I don't want to take any more from Claire's work. I won't describe it any further, but that'll be the topic. And I guess you can judge for yourselves. Is that bold and pioneering as outlined by Dr. Bateman? So Claire, thank you very much. Please come up. We'll get the, we're going to give you the plaque now. Which right here we need like an official sort of photo. We'll do this now with this right here, with the handshake and the photo. Very good. All good? This does have a package to it.
Oh, right.
You can,
All right. Let's see. Thank you very much for having me here today, and thank you to the selection committee, Randy, Tim, Jinmu. I am very honored, and I'm really honored, particularly for what Randy mentioned. This is honored and humbled and a little scared. This is an award for something I have not yet achieved, and so that is a pretty scary thing to accept. And I really hope that we are successful in this effort that I'm going to tell you about today. So my lab at UCSF has one mission, and that is to cure neurodegenerative diseases. And I put this up here as a mission statement, always to ground myself when I'm telling others about our work, that we have a very high bar set for ourselves. I think it is possible in the current era. And I do not know that we will actually achieve this, but the goal is really to be able to tell our patients, particularly gene carrier patients, on the worst day of their life, that they think they're the most unlucky person that they inherited mutation, that actually you're lucky you got that mutation because we can fix that. The time is really right for gene therapies. CRISPR is revolutionizing science and medicine. I don't think we have felt even the beginning effects of this revolution, but it is happening. When we have traditionally considered rare diseases, we've considered them individually, but as a class, they are a very common form of disease. 41% of infant deaths are genetic. We all carry an estimated 2 lethal mutations. They're not lethal in us because we have maybe a heterozygous form of the mutation and it would only be lethal in the homozygous case, but we all have lethal mutations in our genome. And CRISPR is a technology that allows us to remove these mutations, fix these mutations, and I think this is something that will affect and treat millions of people worldwide. I'm not going to talk so much about the CRISPR today. I gave a talk at noon that was a bit more on the CRISPR work, but I'm going to talk about one of the major reasons that is delaying CRISPR from entering the brain and spinal cord and treating our neurologic patients, which is the problem of delivery. So we already know what we can do for many diseases, and technically treating with CRISPR is not that challenging, except that we can't get it where it needs to go in order to treat patients. And I call this the problem of human delivery. I don't need to tell folks here at the epicenter of genetics for dementia, but genetics are a major contributor to dementia. We have rare diseases that cause, rare mutations that cause rare diseases. So this is a picture of Lindy Jacobs and her family from the New York Times that's posted here with permission about her mother's fight with FTD that is caused by a MAPT mutation and her own knowledge that she carries the mutation and is trying to secure her future. As well as common mutations like ApoE4 that maybe aren't the direct cause of disease but are a major risk factor disease and account for many folks around the world. I think a lot about disease and how therapies are developed historically for them because I want to learn from success cases so that we can treat patients with neurologic disease. And I think there are really 3 ways that drugs are successfully developed, and you can argue with me about this after the talk. The first is serendipity. So someone boiled some bark and we found aspirin and we found that it was really effective. I think actually a lot of what we do is still in the serendipity category. And some of the work I'm going to talk to you today is about trying to employ serendipity. The omics work we're doing, the fishing expeditions, this is really relying on if we just do enough, we'll just find it. It'll just happen. The other way that drugs are successfully developed is for genetic disease. We can think about statins, which were developed for familial hypercholesterolemia, but have a broad implication for the general population, but really developed for genetic disease where nature has done the experiment and shown us the reduced mechanism or path forward. And finally, technology. And I think a lot about vaccines. So the first vaccine discovered in the late 1700s wasn't platformized and applied to other diseases till the 1950s. Why is that? Because we lacked the technology to grow viruses. It wasn't until we could grow a virus in a chicken egg, right, that we were able to develop the polio vaccine, and the rest is history. And Sidney Brenner, I think often about Sidney Brenner's quote that progress really depends on techniques or technologies, then discoveries, then ideas. And I have thought deeply about this because why haven't we yet cured Alzheimer's disease? I think some of the most brilliant people in the world have worked on this problem. And if they have not solved it, when I started my career, I thought deeply about this. If Dave Holtzman has not cured Alzheimer's disease, then there's something else fundamental that we are probably missing that we need to think more broadly about. So CRISPR is at the epicenter of genetics and technology. That is why it's so revolutionary, and that's why I think it's so powerful, because it's going to leverage the natural human experiment of genetics along with new technology that allows us to rewrite the genome. I think about CRISPR therapies in two components, and you have to put these components to have a successful therapy. One is the guide RNA and Cas enzyme and how you edit the genome. Again, I said, it's not trivial, but it's not really all that hard. It's programmable. But the second half of this equation is what I'm going to talk about today, and that's delivery. How do we get CRISPR to the target organs? And this is what is the bottleneck in the field broadly. So CRISPR gene therapy, just to give you a high-level overview of what I think it will look like, patients will come to clinic, they will get sequenced. From that sequencing, we'll find their causative mutations that I've depicted in red, and also information about their surrounding single nucleotide polymorphisms. That's differences between your paternal and maternal alleles. And my lab generally targets those mutation, those single nucleotide polymorphisms and cis with mutation to remove the gene-causing mutation from the genome, leaving one copy intact. And we do that because I figure that we don't know everything about genetics and the function of every gene in every cell, and that genes are probably have important physiological roles. The cell then repairs the double stranded break we made and removes the mutation from the genome. Okay. And to let you know where we're at with C9orf72, the leading genetic cause of both FTD and ALS, we are testing our lead therapy in mice. I'm not going to talk about this work today, but I just want to give you an overview of where these programs are at. We have a program for genetic FTD, for Alzheimer's disease, and an early program for Parkinson's disease. So these are moving quite quickly, even through the preclinical pipeline in an academic lab. But again, we don't have a therapy to put in patients because we don't have delivery. So let me talk to you a bit about our delivery plan. The first, fastest route to patients is to adopt an AAV. We have chosen to use Ben Deverman's humanized AAV. So this is an AAV developed to cross the blood-brain barrier by leveraging the human transferrin receptor. And he has shown previously in this publication in Science that he can get broad brain and spinal cord coverage. So she can basically hit the entire brain and spinal cord. And Tan Wang in our lab has adapted this AAV for CRISPR. We use a dual vector AAV. And in a Porter cell line that only changes color, turns on a red fluorophore after it's been edited, she has shown that we can also achieve whole brain CRISPR editing. This is quite transformative in the field. But AAV is not the right tool for CRISPR. We're still using it because I have patients, they are dying. We want to get something to patients as fast as possible. But AAV is really not the right tool for CRISPR for two reasons. AAV is well designed to continually express its cargo or protein that it encodes. But for CRISPR, we want CRISPR enzymes, the Cas enzyme, to go in, do its job, and turn off. If you continually express a Cas enzyme, you risk increasing off-target effects, so genomic damage over time, that I think will be very deleterious, and immunogenicity effects. So eventually, I think if you left CRISPR on in cells indefinitely, eventually that cell that you tried to fix may die from the CRISPR side effects. So what we want is transient delivery. These are the delivery particles that I think have the fastest route to clinic and are viable in terms of both delivering CRISPR and manufacturability. AAV, again, is going to be the fastest to clinic, but probably the wrong tool for the job. And then transient delivery vehicles like lipid nanoparticles, viral-like particles, which are essentially empty viruses that we can load with mRNA or protein, or the naked protein itself, and using protein conjugates to try to distribute CRISPR to the brain and spinal cord. So there's a lot of options. The lab works on all of these options. We also collaborate with companies and academic individuals around the country who are also working on the delivery challenge. But we have a second problem, and that is how do we figure out which delivery particles will work? The human brain is complex, we know that. But it is not just a larger brain compared to a non-human primate or a rodent. I drew the human brain, a macaque brain, and a rat brain to scale, and I couldn't even fit a mouse brain on this slide because it is very small. The human brain is bigger by mass, and for most of drug development, we have treated the brain as just a scaling problem, an engineering problem, that you need to take some drug and get enough of it into the brain. But the human brain is also very unique, and the delivery challenges teaches us how specific it is to our species. We have AAVs and lipid nanoparticles that work in mice and even subspecies of mice that don't translate to non-human primates. We have drugs that work in non-human primates that don't translate to human. And particularly I think this is a big challenge for the blood-brain barrier. So I think this is one of the reasons that therapeutics for the brain have lagged behind other organs. It turns out, CRISPR's in the clinic already for blood disorders, but also for liver disease. It turns out the liver in a mouse and a human is pretty similar. It does a pretty similar job. The proteomics are pretty similar, but the blood-brain barrier between a mouse, a non-human primate, and a human are all different. And if one species does not predict the effect in another species, then it'll be very hard to determine what will work in humans. So I have described here the current translational pipeline. It's busy and it's supposed to be. You don't need to look at every individual word here. But essentially, I argue that our current translational pipeline or discovery pipeline from some idea you have all the way to clinical trial is built to find failures. And that is because that is what it finds. The number one outcome of clinical trial are things that do not work in human. So the current pipeline is you have an idea in the lab, you test a bunch of things, you come up with some sort of lead candidate something. You take that back to animal systems, rats, mice, non-human primates. You screen through the animal system. You eliminate a lot of things that don't work in the animal. You come out with a lead therapeutic that you then take through tox studies, typically in rodents and non-human primates. And the first time that your lead candidate therapeutic or diagnostic ever sees a person is in clinical trial. And for therapeutics, that clinical trial is for a patient who is hoping for a benefit. And often what we find at that stage, after millions of dollars and many years of investment, is that whatever we had didn't work in the first place. So my group and many folks who have joined us on this effort are hoping to revolutionize the way that we conduct this sort of research by going straight to the human case at the discovery stage of research. How are we able to do that? We are planning to utilize brain-dead patients for discovery-based research. So these are folks who have died from a catastrophic injury or illness, something like a car accident or a stroke. They are dead, but they are still in the ICU, hooked up to the ventilator with perfusion. This is the typical source of organ donation. So folks who are going to be organ donors are often pronounced brain dead first. So they're dead, it's not a clinical trial, and since they're not alive, it's not a clinical trial, we can do discovery-based research-grade research. So we're planning to screen delivery particles in the human body to figure out which particles go to the brain. And this work is really also enabled by barcoding technology that has advanced over many years to allow us to do hundreds to thousands of studies in a single subject. So the way this works is we take AAVs or lipid nanoparticles or peptides. We have developed barcoding strategies for all of these different modalities. We barcode the particle with a composition of matter. So it's a tag like a barcode that you would use at the store. We barcode the composition of matter and the route of administration, so how it got into the body as well as what it was made of. We then release all of these particles. So we haven't done this yet, but we're gearing up to do that this year. In the first instance, we're going to inject through the IV as well as intrathecally. So we'll release these particles, and I consider it like a horse race. Which particles made it to ultimately the spinal cord? Because the brain is injured in brain death, we'll use the spinal cord as a readout. We think that's a good surrogate readout because all the available animal model data suggests that if you find something that works really well in the brain, it also works in the spinal cord. And the spinal cord is typically well perfused and intact, even in brain dead patients. And if we are able to successfully do this, I think we will revolutionize the way we do discovery research. And so the discovery pipeline and translational pipeline to clinic will look something like this. For us, it's delivery particles, but I can see that this would rapidly translate to other areas of human discovery biology research. We will take our particles, we will put them into a human brain-dead cadaver or a physiologically maintained deceased cadaver. From that study alone, in a single body, we should find things that we know will work in humans. So we already know it's efficacious or valid. We take them back for limited preclinical testing and toxicology in animals, for which animal models are actually good predictors of toxicology. And then we move on to the clinical trial. So this could potentially compress a pipeline that takes decades or longer into months to years. I will say that we are not the first to propose using brain-dead cadavers or physiologically maintained deceased for research. Very briefly, the first, there's probably more research than has been published, but the first research that we could find was using esophageal airways, which are precursors to the modern intubation technology, was first used in a patient who was recently dead. So not a brain-dead physiologically maintained cadaver, but someone who was recently dead. Folks used that technology in the 1980s to de-risk it before going into a live human. The precursor of TPA was also tested in a recently deceased patient. In the 2000s, 2002, folks tried to map the vasculature of the liver in a brain-dead patient using phage display. And more recently, you may have heard of the porcine to human transplant studies were also de-risked in brain-dead cadavers or physiologically maintained deceased before going into human. That is very late-stage research, so that's kind of the final stop on the train before you get to a clinical patient. What we are proposing is to invert this and be the first stop on the train. We published our proposed work and the ethics surrounding this. We have spent a lot of time thinking about this platform and the implications, the ethical implications of this work. We settled on a terminology, physiologically maintained deceased, and I am open to opinions about what the best terminology is. We want to recognize that folks are dead, but it's not the typical cadaver that's in your autopsy suite. This is something a little bit different. And I would like to acknowledge Neil Single, who's a critical care physician neurocritical care physician at UCSF, who from the moment I told him this idea was on board and has helped me be a true partner in this work, helping get the IRBs through our institution and working tremendously on the clinical side. We have also done ethics work with Doug Pett, who is a neurohospitalist, and Brendan Parent, a lawyer who I think is one of the leading neuroethicists in this space on the use of physiologically maintained deceased. So this is someone who's died by brain death criterion, so they're pronounced dead, but as I mentioned, they still have physiological support. We talked about some of the benefits already, that the best data that we can get comes from humans, and this is the closest we can get to a living human study, in my opinion, without using a living human. It's expensive to do up front, but I think ultimately down the road it could be very cost savings and mitigate our timelines, our failed studies, and the risk to patients. But there's a lot of risks. One of them is the way we treat deceased folks. And we've worked through with our IRB and our institution to make sure we mitigate as many of these risks, but I just want to acknowledge that they're there. The second is that we potentially would detract from the care of living patients. And the biggest risk is that we undermine public trust. The number one reasons that you probably can guess, the number one reason that people don't donate their organs now is that they're worried that the doctors in the hospital will hasten their death to get the organs. So that is a real problem for us as a medical community, maintaining that patient trust and maintaining the organ donation program, which is so life-saving for so many folks. So some of the ethical principles we have decided on is we will not use a cadaver that can be an organ donor. So if they're eligible for organ donation, we will not take the life of someone who is living in order to do a research study. So these will be folks who are potentially wanted to be organ donors but are not eligible for organ donation. And that's not as uncommon as you may think. We think there's several a year, and we've worked with our organ procurement agency, which is a regional network that manages the organ donation program. They actually screen everyone, and they have folks that may be eligible for these studies. We have agreed that the ICU should not be full when we are doing our study, and someone's not waiting for a bed in the ICU to get life-saving care so that we can do research. And most importantly, we've engaged the stakeholders in the hospital, medical ethics, stakeholders in the community, folks who would be impacted by this work to make sure that everyone has a voice in how these programs are run. I talked mostly about ideas today, and I just want to leave you with a tiny hint of science. I talked about the particles that could potentially deliver CRISPR to the brain and spinal cord, and I just want to give you a flavor of some of the work we're doing on lipid nanoparticles. We in the lab, Ranbir Chow and Greg Moll, have done screening along with Sonhae Im on developing lipid nanoparticles and testing them across cell types that differ. And this is a small screen of 20 lipid nanoparticles for which we're delivering an mRNA fluorophore. So the cell has to turn on the mRNA that we deliver in order for us to see it in the cell. That indicates that it was successfully delivered and that the cell machinery is still intact, the cell's alive. And you can see right off the bat that there are different lipid nanoparticles that work in iPSCs versus lipid nanoparticles that work in postmitotic neurons, and that these don't overlap very well. Sanheim went on to test lots of different cell types. We've tested iPSCs here on the left, neurons in the middle, astrocytes, and again, we find this cell type specificity that's incidental to the lipids that we're including in these lipid nanoparticles. And we have screened over 200 lipid nanoparticles across many cell types from different species, human-induced postmitotic cells, as well as mouse cell lines. And again, we get maps of lipids that work in different cell types. We are also recapitulating that the mouse data does not really predict the human data, even in the in vitro state. So this lipid nanoparticle library, which has got fancy barcodes, is being built and actually will be deployed in a brain-dead patient hopefully sometime soon. Okay, so we're delivery agnostic. I just want to find the thing that works the fastest, and I think it's a numbers game. I think I want to employ that serendipity. If we could just test enough things, I think we can land on an answer. So we built these systems to be high throughput so that we can scale rapidly. We are testing all the available technology, but we're open to new technologies as they're developed, and this platform is really built with future-proofing in mind so that new technologies can be rapidly de-risked in the future. We test them across in vitro cell types so that we know things will work in the human cells that we care about. We do some animal testing, really for route of administration, and because the current FDA pipeline still requires some efficacy testing. And then we're really using the human platform to de-risk right from the start of the research what will work in humans in the future. So our goal is to bring CRISPR therapy as quickly as possible to patients for potentially life-saving therapies. We are focused on human tissues out of necessity. I think that if animal models don't predict what's going to work, we have to find alternatives to the traditional way of doing science. And if we do, I think this will change how we do biology, discovery-based biology broadly, not just for this specific class of translational delivery. We're working to develop first-in-class genome therapy, starting with C9orf72 FTD ALS, but also we have programs for FTD and Alzheimer's, as I mentioned. We try to remove the mutations from the genome. I didn't talk about the biomarker and bioinformatic work we do to operationalize all of the work that we're doing for future clinical use, but hopefully this will be a roadmap for how we can perform discovery research broadly and bring new therapies to patients. So Randy mentioned we had a conversation about what it means to be a bold pioneer, and I give a lot of thought to this. I think that, and I wanted to share a couple of my favorite quotes. One by Eleanor Roosevelt, who we all know, but who was so instrumental in developing the International Human Bill of Rights that is now, being questioned. And I think a couple of the things that I appreciate about other leaders, talking about all of the great leaders throughout history across all disciplines, I think people are driven. They have some sort of internal drive, something to prove and contribute to the world that's very meaningful to them. I think that people are creative, and I have often found in my life that some of the most creative periods are the times you're new to something. So, right, I was new to delivery research. This is not something I trained in. This was a problem that needed to be solved. When I came up with this solution, and I think my training in neurology really helped guide that, like, why don't we just do this in a human? We can do this, we can try this. But I highly recommend getting involved in new situations, putting yourself in uncomfortable learning situations where you're not the expert, you're not the most talented person in the room, so that you have an opportunity to have fresh eyes on a problem and contribute. I highly value the most junior people in my lab because they are the ones that are as free from the cumbersome details of how things have traditionally been done to think of new ideas. And I think there's an exercise that I do for any problem that I will share with you that I do for any problem that we are thinking about taking on, or that I personally am thinking about taking on, which is I go through a series of questions in my mind, and if I meet all the criteria, then I'll take this challenge on. And the first thing I ask myself, is this the most important thing I can be working on? Because life is long, but it's also short. And then I ask, even if it's the most important thing, is it the right time in history to answer this question? Because there are things that I would absolutely love to do, but we just don't have the tools yet to do them. I would love to solve consciousness, but I don't even know how to approach that problem. I then ask if we have the tools to address this question. So for instance, it would be great to treat genetic carriers of disease, but before CRISPR, was that really possible? The next question is, if we don't have the tools, can we make them? And this is where my lab often comes into play. So it's yes to everything. The tools don't exist, but it's possible to get them, and we just don't have them. Then we spend time trying to make the tools. And only then do we take on a challenge. I think often we're trained to think small, to take on small challenges, to differentiate ourselves, to find something that your mentor didn't do. And I got the advice relatively early on, you can do that, but you can also do the most important thing that you can spend your life doing. And it's not trivial to think about and decide what the most important problem is. And so I recommend that you spend time really thinking about that. I do that all the time, and I still reevaluate at every stage whether I think we're working on the most important problem in the world. And I think you should not be shy. I was never trained to do CRISPR. I did a PhD in animal behavior. I did a postdoc in something else, and I looked around and said, CRISPR is going to change this landscape. I need to go train in that. You can change at every stage and do something if you think it is the most important thing that you can do with your time. And so if you know this quote by Toni Morrison, if there's a book you want to read and you can't find it, go write it. I think that applies to science too. So with that, I would like to thank the committee for bringing me here and for this amazing award. The lab who makes this possible, obviously none of this is done in a vacuum. Team science is the norm now. We would not be able to even think and dream of these projects without multiple stakeholders being involved, including the lab pictured here. Our collaborators, Neil Single, has been a tremendous force in helping us really translate this to clinic. My chair, Andy Josephson, has also been very supportive. The neuroethicists, the collaborators, Ben Deverman particularly, our funders, and most importantly, the patients, who are really the driver of this work and the mission that we are trying to accomplish. Thank you.
You have time for questions.
Thank you, Claire. This talk was amazing, just like the one at noon. I have a practical question. I'm also a huge fan of being bold and trying new things. But what advice or what has worked for you in actually getting funding to do such a thing?
So I will say, I said at the beginning, standing up here and giving a talk about something I have not done has been really scary. But the best advice I have is to put your intention out in the world. Because we do have funding. So we have been funded, which is kind of unheard of, by the NIH to do something that's never been done. Usually you have to have a lot of feasibility data, et cetera. But put your ideas out there, stand behind them, get the feedback, and other people may buy into your vision and support you as well. So that's why I said this is a team sport. This is not me up here just, oh, wouldn't it be cool if we could do this? I mean, I cannot thank the NIH enough for the work that they did to get us funding, to create a pathway for something like this to be funded, and to standing behind us even with all of the chaos that has happened in the last year.
I think it's really clever what you're doing. And I was just curious, when you find what you're looking for, what's the next step? Like now you find something that targets the spinal cord and the brain, because what do you do? How do you take that and say, how is it safe? Can we put it in a person? Like, how do you move beyond the cadaver?
We only have to go one step back, which is to do toxicology in animals, and then we think we can go to clinical trial. And I will say that we built this for the brain and spinal cord. I didn't even tell you the major mission of this effort. We built it for the brain and spinal cord because that's what I care about solving, but we should be able to solve the delivery problem for every viable organ in brain death. And the plan is to test heart, liver, kidneys, spleen, et cetera, get the answer for everyone. We will hopefully share all of that with the world so that other folks can go make the therapies for those organs while we focus on the brain. But I think if this pathway works, it becomes a very quick pathway to clinical trial. We've already done a lot of work on the CRISPR targeting. I'm not going to minimize that. We're going to add that to our particle, and then that goes into toxicology testing and hopefully into clinical trial if it's safe.
Claire, that was a fantastic talk. Really enjoyed it, and a bold idea too. A question, so with brain death caused by a catastrophic injury to the brain, I like how you're using the spinal cord, but are you concerned that particles may enter the brain and go to the spinal cord without crossing the blood-brain barrier? Is there a way to check for that?
I think it's possible. We should be able to see a distribution throughout the spinal cord, which will give us some confidence. We are working on non-brain dead, non-ICU models of human tissue to be able to answer some of these questions more rapidly. They're not quite ready yet, but I think brain dead is the first step. It's obviously very expensive and hard to do. If we could do these sorts of tissue studies in a rapid manner, I think we can even accelerate these timelines. So I think there's other ways to de-risk the brain part of this platform that will come down the road.
Claire, thank you for the talk. So a technical question, how can you make use of LMPs in a targeted way to affect only a specific cell type of your interest?
Okay, so the lipid nanoparticle so first of all, I don't take the position that we need cell type specific targeting. There's a lot of folks working in the field on how do we get this AAV into this specific cell type. I think that is all basic science interest for mapping and neural mapping, et cetera. Therapeutically, there are not very many diseases that are cell autonomous or that we can prove are cell autonomous. A lot of diseases have multiple cell types. So I take the position that we should try to hit as many cell types as possible for therapeutic benefit. Just like I don't know all the functions of a gene in a cell, I don't know all the cell types involved in a disease. So we are not trying for cell type specificity. This has been an incidental finding of the work of doing lipid nanoparticles, that the lipids themselves seem to have cell type specificity. Now that is sort of mind-blowing biologically that I think we should spend a lot more time thinking about, but it's a technical engineering hurdle that we will need to overcome. But we will not know till we've really profiled. The lipid nanoparticle space is almost infinite. So there's 4 to 5 components of a lipid nanoparticle, and within those there's a lot of room to design. So you can essentially design anything that you want in that space, and that's without even protein targeting. My lab currently is working on, we can decorate those lipid nanoparticles with proteins, we can further refine the delivery specificity. So the goal here is to not get cell type specificity. We want something that hits multiple cell types. I think what will happen is we'll have a menu of options that you want to hit this organ and this cell type, and you're willing to hit these other cell types. Here's the things that will work for your project that we will choose from. And we can also combine lipid nanoparticles, right? We'll need a neuron, an astrocyte, a microglia one for this therapy. That is down the road, I think, the goal. But right now, we just need to map the space.
Thanks for that really inspiring talk. I think you obviously were a great choice for this because you really embody the bold spirit that people are looking for. Coming from the opposite side of the world, being a timid sort myself, I'm just curious about how you're thinking about the N you're going to need to move to a next step. So, I mean, men and women are different. The brain and spinal cord are really different. These are not well tissues because, there has been some massive injury that has made them non-viable in the long run. And so it strikes me that you've got a lot. I mean, this is really risky. And I wonder how many times and how many sort of, what age, young people and old people are really, really different in the characteristics. You can move things in and out of their brains. And so how quickly are you imagining you can go from observations in relatively small numbers to an experiment in people?
Yep, it's a good question. I would feel comfortable moving forward with an N of 1 if it looked really good. I still think it's better than the animal models. So it's, another kind of way I think about clinical trials is amyloid lowering therapies. How many clinical trials did we do before we got to something that seems like it has efficacy? I don't think that was because they were poorly designed, et cetera. I think it was because that first human experiment was the first real experiment. The mouse really didn't inform us. And it took many iterations. And I'm so grateful to the entire biomedical community that didn't give up and continued to push that therapy forward after many iterations. But to answer your question, there's absolutely going to be differences between individuals, et cetera. I did not build this to find subtle differences, to test sex differences, et cetera. I found this to find home runs. So I think home runs are likely to work across multiple people. I can't say that for sure, but when we do 2 people, I think we're going to have pretty good indicator that they work. There's going to be a lot to learn over time about all these nuances, but right now we just need the best performing particles. And the animal studies really show us that we can identify those in a single animal. You can repeat that across mice or non-human primates, and the answers still come up the same. Now, if you compare a mouse to non-human primate, totally different efficacy. But if you take several non-human primates, the lists are relatively similar. So we're not looking for did the one that reached 10%, was it 11% or 12%? I want the one that reached 90%, and I think it's likely to work. The second half of your question. Tell me again. It is risky, but we got to try. We'll find out.
So I think this question was maybe already asked. How are you planning to differentiate uptake from productive uptake in this test?
Yeah, it's a really fair question. So one of the main problems in delivery is that you get your particle somewhere near, but it's not in the cell, it's on top of the cell, or it's in the blood vessel, and how do you differentiate that? So we have functionalized all of our barcodes. I didn't talk about that scientific side today. The barcodes, particularly for lipid nanoparticles, have to get, like the GFP that I showed you, have to get into the cell and be expressed, actually, for them to turn on. Our barcodes are nuclear localizing, but the mRNA gets in, it's translated, and then the barcode is dragged into the nucleus. So, and then we can do single-cell transcriptomics or spatial transcriptomics to really read out the cell type specificity. But it's all in the barcode functionalization. We won't be able to see the particle, it'll be gone at that point. And even if you could, like using dyes, et cetera, it doesn't actually give you the functional data that you need.
Okay, maybe we'll leave it there. Let's thank Claire for another. Oh, Randy had one question. Burning question from Dr. Bateman. Then we'll close it out.
So Claire, you talked about the pearls of wisdom, the positive things, the looking for the home run, the big magnitude effects, finding what you're trying to accomplish as a life mission. And I do agree all of that's really important. What you didn't mention so much though was the challenges and the barriers, the things that almost stopped you. And I'm just curious, do you have any thoughts to share with the group about this? And how did you handle it? How did you think about it? How do you manage those things that sometimes might almost stop you from doing what you believe in?
I meant to mention that. I was going to say the other thing that I think people who really do transformative work have in common is they have experienced profound failure in some capacity. I think there have been a few times in my life, and we can talk about it another time, where I've experienced the kind of failure where you have to soul search about who you are and what you are doing. And I think that is very important, because if you can build back from that, you can first of all, realize that you have not much to lose, but also you become kind of unstoppable from the noise that continually follows around people who are trying to do things differently. So whether that's failure because of life circumstances out of your control, or failure because of things that have happened to you or things that you've done, I think actually failure is really important, and it's not comfortable going through it. It's really challenging. It's about whether you can survive it and grow from it. The challenges are many. I think I talked to my mentors. Randy's helped me a few times. I continue to have challenges. The challenge is, once you have honed in on the thing you need to do and you feel confident that you have a mission that needs to be accomplished, I think the challenges become noise outside of that mission, and you weigh whether that's a challenge that you're going to allow to stop you or not. I will say plenty of people will try to stop you. And maybe you'll try to stop yourself. There's definitely times where I've thought, gosh, there's a lot easier things I could be doing. I could have my 1R01 in my lab with 3 people, and I could go on lots of nice holidays and be home every night, having dinner. Or I could try to do this mission-driven work that is very time-consuming and takes my heart and soul. I don't have all the answers. I don't know that I'm very good at it. I will say that things bother me a lot. I still get worked up about things. I'm not I try to do that behind the scenes. but it still happens. You will have challenges. You will have failures. You just need to have more successes than failures. And if you look at professional athletes, they don't hit a home run every time. They have a lot of strikeouts. They have a lot of lost matches. And that grounds me and helps me to keep going.
Seems like a fantastic way to. Thank you again, Claire. That was fantastic. Brooke and Tracy have put together a very nice reception for us just outside. I think it should be ready now, Tracy? Yes. Ready to go. So we'll see you all out there. Thank you.